All right, I would like to thank the network for inviting me and for supporting this project that looked at the impact of stress exposure in a spontaneous mouse model of breast cancer, the MMTV-PYMT mouse. And so the central questions for this project was, does stress alter tumor progression in MMTV mice, which are a spontaneous human mimetic tumor model? Does stress alter circulating exosomes in this model, and what stress pathways underlie changes in both tumor progression and changes in circulating exosomes? So to briefly talk about the MMTV-PYMT model, this is a transgenic model in which the oncoprotein polyoma middle T antigen is exclusively expressed on mammary epithelial cells, resulting in mammary tumors in the mammary glands over time. Now, in this mouse strain that we used, they're on the FEB background, and the progression of the mammary tumor development is fairly quick, and they begin with hyperplastic lesions as early as 4 to 6 weeks of age, and advance by 8 to 9 weeks of age to advanced pre-malignant lesions. By 10 to 11 weeks of age, they're in early carcinoma, they're invasive, and by 13 to 15 weeks of age, they are late carcinoma, and they are metastasizing to distant sites, and specifically in this model, it is to the lung. These mammary tumors are hormone receptor expressing. They express estrogen receptor, they express progesterone receptor, and it is an immunocompetent model, so these mice have a fully functioning immune system. And we developed what we're calling a dual stressor model to conduct this study, in which mice are socially isolated and then exposed to a short period of an acute restraint stress. And we developed this simply because in earlier studies, using simply social isolation, we were seeing no changes in tumor progression. So we realized we had to do something a little bit different. Looking at the literature, it was clear that imposing an additional acute stressor elicited very nice and particular corticosterone responses. So that's sort of why we developed this. And again, just briefly, the mice arrive and are group housed at around 6 to 7 weeks of age when they're in the hyperplastic pre-malignant phase. Around 2 weeks later, we begin stressor exposure. And in these experiments, I'm going to show you 2 experimental groups, non-stressed and stressed mice. The non-stressed mice remain group housed throughout the experimental paradigm. The stressed mice are transferred to single housing sort of at the beginning of the malignant transition. 2 weeks later, we expose them to 3 consecutive days of restraint stress, which was 2 hours per day, always at 9 o'clock in the morning. And after each restraint session, the stressed mice were returned to their original home cages. We looked at 3 different time points, sort of representing important points in tumor progression: 24 hours after the final restraint stress, or 2 weeks after, or 3 weeks after restraint stress. And first, we wanted to sort of characterize what the stress hormone response was like in these mice. And when we looked at spleen norepinephrine, norepinephrine is a metabolite of norepinephrine, so we're using it as a measure of sympathetic nervous system activity. Immediately, let me back up first. In all these figures, blue represents the group-housed non-stressed mice. Red represents our stressed mice. In these initial experiments that I'm showing you, we've got an N of 14 to 16 mice per group, and that's what the power analysis said we needed, and it's absolutely true in this case. With sympathetic nervous system activation, we saw that readily at the 24-hour time point, that's 24 hours after the restraint stress. The effect was not apparent at 2 weeks. after restraint stress, but it was sympathetic. We did find evidence of sympathetic nervous system activation at 3 weeks post-restraint stress, and we noted, although I didn't investigate any further, that the issue at 2 weeks appeared to be actually elevation in norepinephrine in the non-stressed tumor-bearing group. Now, when we looked at HPA axis activation in the form of plasma corticosterone, dramatic elevation in corticosterone throughout tumor progression. And again, 2 weeks and 3 weeks after we have given them the restraint stress. Those mice are still socially isolated, but that's a rather dramatic elevation in corticosterone. And in fact, as an aside, in the FEB mice, in non-tumor-bearing FEB mice that undergo exactly the same protocol, the plasma corticosterone response is completely at baseline starting at 24 hours. So I just want to point that out, that the stress response in these mice is actually quite abnormal, and it's something we'd like to investigate a little more. And in these mice, again, tumors develop in all 8 mammary pads, so we simply, to assess tumor growth, you harvest all the solid tumors and weigh them, and you generate a number that's called total tumor burden. So again, in our non-stressed group over time, shown in the blue bars, with a progressive increase in tumor burden. And rather unexpectedly, in our stressed mice, tumor burden was reduced, particularly at the 2-week time point, but even 3 weeks after restraint stress. And this goes very much against the current literature. And so we also looked at lung metastasis as measured by just H&E stained sections taken throughout the lung. We just total the number of lesions that we detect. In this particular experiment, at the 2-week time point, we had a relatively small frequency of mice where we could even detect metastasis. When we just included those mice in this graph, there was no change in lung metastasis. But probably at the 2-week time point, the frequency is probably more important, and there was no difference in metastasis at the 2-week time point. At a week later, when 100% of the mice had detectable metastasis, similar results, we had no change in the number of lesions we were detecting in these mice. Again, a little bit surprising. In these mice, we were collecting a lot of information, and one of the things we were looking at is a couple systemic responses. You can see my poster for other things that we measured, but here I'm going to focus on circulating exosomes. We were really interested in using exosomes as potential biomarkers of the stress response. And so let me tell you a little bit about exosomes. They are nanometer in diameter sized particles that are sort of a relatively newly described means or mechanism for cells to talk to each other, both this happens at a local level. It also impacts, they can communicate distantly. And this is an important, relatively newly described mechanism, particularly of setting up metastatic lesions. So exosomes are they're one of a large, one of the components of microvesicles. And so in the microvesicle world, you sort of want to characterize these particles because they are very, very small, and they could be confused with protein aggregates and things like that. So in our case, the exosomes were isolated from plasma using a commercial product called Exaquick, and we found it did a good job of isolating what we are calling exosomes. And it's based in part on their size, and that ranges between about 50 to 150 nanometers. And this nanocyte video that I'm actually not going to run here, I'm going to show you some data that shows you specifically the sizing on the particles we isolate. And I want to point out that exosomes are released by all different kinds of cells. They're released in health and in disease. So they are very, very interesting ways of packaging DNA, RNA, and protein and taking it to other cells that take it up. And let me just mention, in the context of tumor biology, exosomes are thought to promote tumor growth, progression, and alter immune function within a tumor. So based on the nanocyte particle analysis, again, we have a very clean population of microvesicles, and these microvesicles, we looked at some exosomal markers. Exosomes are thought to derive from the endosomal membrane, and there's some sort of canonical markers that are included here that this population of microparticles expressed. They were negative for a marker of cellular contamination. That's a Golgi marker, GM130. So we looked at a number, we only analyze proteins in our exosomes. We looked at a number of cytokines and chemokines that are relevant to tumor progression and found no difference between stress and non-stress mice. However, we noted that TGF-beta was particularly a predominant protein within these exosomes from the tumor-bearing mice. And we used a multiplex analysis to separate TGF-beta 1, beta 2, and beta 3 isoforms. And what we found was a significant reduction in TGF-beta 1 containing TGF-beta 1 concentration in exosomes at 2 weeks, which was gone by 3 weeks. TGF-beta 2 content was also significantly reduced 2 and 3 weeks after restraint, and a similar pattern was detected with TGF-beta 3, although it was at very low concentration. So this result actually mirrored our changes in tumor progression. We next wanted to determine what stress pathway might underlie these changes. And in this experiment, we tested whether stress was acting through the sympathetic nervous system and specifically through beta receptor signaling to inhibit tumor growth and progression. And here we used Nadolol, which is a non-selective beta receptor antagonist. It does not cross the blood-brain barrier. So we use this to block peripheral beta receptors. In this experiment, also, we did not want to be picking up and injecting these animals every single day, so we used a subcutaneous continuous release pellet, which was provided 3 days before the start of social isolation. And these pellets were on board and blocking beta receptors throughout the stress paradigm. The controls were implanted placebo pellets. So this is a 4-group experimental design. And one thing I need to point out was because we went to 4 groups, we are at an N of 7 to 8 in these experiments. And therefore, in this experiment in which we tested beta blockade, there was a non-significant reduction in primary tumor burden. Again, that's, we believe, due to weak power. But when the animals were pre-treated with the beta blocker, if you just look here at the Nadolol-treated stressed animals, you can see that the tumor burden is equivalent to that of the non-stressed placebo controls. So it looks like the beta blocker did prevent the stress effect. Nonetheless, we also saw in this experiment, the statistical analysis revealed a significant main effect of Nadolol treatment. And in the non-stress group, the beta blocker alone elevated primary tumor burden, and that was not expected. In these mice, we also looked at lung metastasis, saw no significant change with stress exposure in the placebo-treated mice. But once again, in our nadolol-treated mice with the statistical analysis, we had a main effect of nadolol. Again, rather unexpected results that I'll come back to in a second. In this beta block experiment, we had been sort of looking for we had looked at a number of tumor mechanisms to explain why we were getting tumor inhibition. And essentially, the only thing that we found differences between our stressed and non-stressed group was staining for cleaved caspase-3, which we use to measure apoptosis within our tumors. And what we found here was that in the stressed group, we saw a significant elevation in cleaved caspase-3 expression, and it was blocked by nadolol pretreatment. And interestingly, here there was no main effect of nadolol. Nadolol in the absence, nadolol in our non-stressed mice did not significantly differ from our unstressed placebo mice. So there's a tumor mechanism underlying the tumor inhibition. And the interesting part that also came out of this experiment was the impact on TGF-beta content within the exosomes. Now, in this experiment, TGF-beta 1 content was not altered by stress exposure. TGF-beta 2 content was once again suppressed by stress exposure, and this effect was completely blocked by beta blockade. Similar trends were noted with TGF-beta 3. So I've shown you some somewhat surprising data that stress inhibited tumor growth and at the same time reduced TGF-beta content in circulating exosomes. And one question I'm sure most of you are asking is whether released norepinephrine can act directly on tumor cells, as has been shown by several groups, and activate, for example, beta-adrenergic receptors on the tumor cells. So another component of this project was just determining whether these cells express beta-adrenergic receptors. We used a cell line called R221A, which is derived from MMTV-PYMT mouse, and using a cyclic AMP accumulation assay, we determined that that R221A cells respond very nicely to a non-selective beta receptor agonist, isoproterenol, and a beta-2 selective agonist, formoterol, but they do not respond to beta-1 or beta-3 agonists. So these cells are expressing beta-2 adrenergic receptors. And then we did an experiment in vitro to look at TGF-beta content within released exosomes. So the R221A cells were exposed to isoproterenol for 3 days. The exosomes were collected and isolated. And in this case, we actually did a proteomics analysis of the isolated exosomes. And what we found was that all 3 isoforms of TGF-beta were reduced within exosomes released by the R221A cells, suggesting that indeed in the MMTV model, the tumor cells can be directly targeted by released norepinephrine, and in fact, that reduces TGF-beta content within exosomes. So our 2 main conclusions from this project were that peripheral beta-adrenergic receptor signaling constrains tumor progression in MMTV-PYMT mice, and that circulating exosomes may be useful as a marker of stress-induced changes in cancer progression. And I'd just like to make a comment that I think in these MMTV mice, the reason for the unexpected effects of nadolol in the absence of stress is that we might be having, as the tumors progress, we might be seeing elevated sympathetic nervous system activation. That's a whole other interesting area, and I can show you data to support that. at my poster. So I would like to acknowledge my collaborators, and especially Ryan Dawes, who was the graduate student who did all this work and really who got us into the exosome world. And I'd especially like to thank the network for funding this work, because this work could not have been done without it. So thank you, and I'll take questions.